A very good welcome to all of my friends, and in this video tutorial we will be talking about the DNA repair mechanisms and why repair mechanisms are important and what are the reasons for making a DNA repair and why we need this repair at all, and what are the agents that are causing different damages in DNA. Now let us talk about that in detail. Okay, now you can see this why is DNA repair is important. There should be a question mark here. Anyways, now we can see in this picture that a damage of a DNA can lead to different pathways, which can lead ultimately to senescence or aging, and finally to apoptosis or cell death, or sometimes even it can lead up to formation of the cancerous behavior of the cell. So a damage can be lethal at the end. So I call it pathological term, and at the end a damage can lead up to the cell death. So that's the end. Now in every all of us never try to be die, right? But we know that we have to die at one day, but still we try to live on. Now cell is also doing this. Cell is also trying to live on in all the time, in all the situations. So that's why cell will always try to keep itself healthy and fully functional, right? Now if I look at the central dogma of biology, we can actually answer this question pretty fairly. Now the central dogma of biology suggests us we are having the DNA, which is carrying all the informations, which is the head department here. From here we produce the RNAs. Now in this case especially mRNAs, so I refer to mRNA in this case. Okay, now in right after that, what we have after the transcription and the translation of this mRNA, what we produce proteins. Now this is the final moment, right? So from the DNA towards the protein, this is the central dogma, and in this central dogma each of the steps are important, because any mistakes during each of these simple steps, not actually simple, they are complicated, anyways, can lead to the formation of malfunctioning proteins. Now these malfunctioning proteins can lead ultimately to the destruction of a cell. Okay, so for example we are having DNA sequences there. Now any during this replication process of the DNA from DNA to DNA, so let me change a color. For example, there are processes like to form DNA to DNA. This is called the replication procedure. There can be mistake in this replication procedure, and as a result of this mistake, we end up with the deformities in DNA. And if there are any deformity in DNA, that can lead up to the dangerous effects, because any change in the DNA level will be further modified in the next two levels. Okay, so any changes in DNA level will be the most lethal of all of these changes, because it not only changes this DNA conformation, but it also changing its future two round, that is a transcription and translation. So any change in DNA, suppose a normal DNA is changing to abnormal DNA, then this abnormal DNA will be transcribed abnormally to the abnormal mRNA, and that abnormal mRNA can again will be translated to finally make the malfunctioning protein. Now problem can occur in this replication step. Problem can occur in this step from production of mRNA from DNA, which is the transcription. So this transcription step can also be problematic, or some misincorporation of nucleotide can also be possible during this transcription. And finally, during the translation, misincorporation of amino acid sequence can also form the chain termination. We know that as a result of some amino acid production, like the codes or stop codons like UA, UGA, UAG, a total protein synthesis can be blocked, and chain termination can happen even though the protein is not told to be stopped, right, in those particular time. So that's why in these three level, this is the translation. So in these three level are really important, because these are the molecular machineries, these are the molecular mechanisms of production of the protein. That means the expression of the gene which is present in this DNA, right? That's why these are all important. Now any problem during all these steps can lead up to the malfunctioning of protein, and finally the destruction of the cell. So this is the basic point, this is the take home message. Now you can see here in this case, any damage can lead up to the nuclear DNA damage, or it can be mitochondrial DNA damage. Whatever type of DNA damage it can be, it finally, if it is unrepaired, you can see here, in this bottom line we are having the unrepaired. In this top view we are having the repair mechanisms. If it leaves unrepaired, then it will lead up to the pathological development like cancer, senescence, apoptosis, and all these things. So finally all it means death. So these are all are the death signals for a cell, and we know if there is a death signal for a cell, there must be the death signal for us, because we are made up with cells. Okay, now that's why we need to go through the repair mechanism. So any problem, if there's a small problem, we'll definitely check it, and if it is can be done by us, if we can fix it on our own, we can definitely try to fix it, right, in any aspects of our life. That's the truth statement. Now here, if they go through these repair mechanisms, they finally go to the metabolism, and finally they can again make all these things right, and finally metabolism will go on in full force, because metabolism is the key process of generating energy inside the cell. Okay, so these are the basic things. But what are the things that can cause these damages? Now if we categorize them, we can have two different things. One is the exogenous damaging components, another one is the endogenous damaging components. Now the endogenous damaging components can give a rise during the metabolism processes and cellular processes like this transcription, replication, and translation procedures. Okay, and exogenous damaging compounds are several things like UV radiation, like gamma radiation, or infrared radiation, like chemical mutagens, which can cause the base flipping or base modifications of nucleotide sequences. So these chemical components, as well as this light or ionizing radiation and non-ionizing radiation, all of this can lead up to the several abnormalities into this DNA structure, either in nuclear DNA or in mitochondrial DNA. Okay, so these are the components. So there must be component for producing the damage. It can be exogenous, it can be endogenous, but finally we need to fix it before going to the next round of our process or life process. That means the reproduction, right? Now if we are having the healthy cell, that means the rate of DNA damage is that equals to the rate of repair. Only we can call it it's a healthy cell. Now this is a very important concept. Look at it. Now if the rate of DNA damage is equals to the rate of repair, that means whatever DNA damage is happening inside our cell, repair can go and fix it, so our cell remains healthy. That's totally justified, right? Now if the rate of DNA damage is greater than the rate of repair, that means the repair is not working fully functionally, and repair is not able to clear or to make the DNA healthy. For this purpose, or as a result, the cell will be called a disease cell, and that cell cannot live many long time. So those cells will be taken on to this apoptosis or programmed cell death. That those cell can go through this senescence, or sometimes it can maybe converted into cancer cells. Okay. So what happens? This is a quick fact. That's up to almost 5 lakh. Here is the 5 lakh DNA modification events per cell per day is happening. So you can see how much modifications are happening all the time. So I am just sitting here and talking to you guys, and all the modifications are going on inside our cell, inside my cell. But my cell is healthy, and it is healthy enough to protect me against all those modifications. And those modifications are not always wrong. Some modifications are wrong, and some modifications are right, which can take those wrong modifications and put it into right track, right? So this is the process of DNA mechanism, and this is repair mechanism, sorry. Now that's why the DNA repair mechanism is really important above all. Now let us move on to the next slide, and in this slide we will be talking about the regulation of the DNA damage. Now you can see there is a balance between two things. One is the DNA repair, and another one is the DNA damage. Sorry, one is the DNA repair, another one is the DNA damage. So these two things, again my mouse is troubling. Anyways, now repair and damage. So there is a relation between this repair and damage. If repair is high enough and damage is lower enough, then the cell will remain healthy. If the repair mechanism is low enough and the damage is high enough, in that case the cell will die. So this ultimately leads to the death of the cell. This ultimately leads to the proper functioning of the cell, right? Now you can see in this diagram, which is clearly illustrated here, now we are having the DNA damage at the central point, and this damage can lead to two different pathways. One is the survival pathway of the cell, another one is the apoptosis pathway of the cell, right? Now if there are a lot more effective proteins are there, like p53, E2F1 and all these proteins, there are a lot of different proteins are out there which are helping the cell to establish this DNA repair systems, or these are the controller or the master proteins which can regulate the DNA repair system, right? So if these proteins are functional inside the cell, so these are efficient to the DNA repair, and as a result the cell will survive, right? But if there is a higher concentration of all this protein and lower concentration of E2F1 or P53 and all these regulator proteins, and if these proteins are malfunctioning enough, then this will lead to the apoptosis of the cell, as a result that this cannot save this cell from being damaged. Okay, so definitely there is a source. This source is IR, infrared radiation here. So definitely will be a source for a DNA damage, whether exogenous or endogenous. And second thing, this what will determine whether the cell is going to live or die is between the balance between the survival and apoptosis. It's exactly between the repair mechanism and between the damage systems. So it's kind of a balance between themselves, and the balance is in a small tiny pinhead. So it's balancing in a pinhead. Now anything can happen in this case. Okay, but always in healthy cell, repair mechanisms definitely go against these damaging systems, and they manages to keep all this DNAs intact inside the cell and defeat these damaging conditions. That's why the cells live healthy. But sometimes these things are disrupted and cell will die. Okay, that's the thing. Now you can see here another picture is there. DNA damage could be there. It can lead to cell death, or it can lead to mutations. Now mutation means sometimes cell death, mutation means sometimes cancers, and mutations, I can tell you, most of the mutations, not all, but most of the mutations are damaging. Very few mutations are good. But most of the mutations are damaging. But again, mutations are very important components of generating variations among cells. So we cannot go against these mutations. So mutations, we need mutation. A cell need mutation to generate or to create the evolutionary advantage by making the variations among the species. So we need it because it's a natural process of having variations. But still, enough mutation or much more mutation can lead to cancer and cell damage and cell death. Now finally, so DNA damage can also lead to different paths like DNA repair systems, or these are the checkpoint systems which can block the DNA damage before the damage happens. That means there are some error prone regions inside the cell, there are some processes which are error prone. In those situations, there are different checkpoints lie between those situations which can control tightly whether the cell is going to cell want to go through that checkpoint or not. If everything is okay, everything is alright, then only the checkpoints open and it allows the cellular process to pass. But if anything made wrong, then the cellular process will be blocked, right? So this is about the cell cycle, checkpoints, and DNA repair system. Okay, now that's how the DNA damage is being regulated with the help of the DNA repair. So it's again, it's a race between the DNA repair and the DNA damaging components. Now let's move on. Now here we can see a lot of different what you can say informations are there. Now there are different DNA repair systems which are available in the cells, bacterial cell as well as in mammalian cells. Now we can find what are the damaging agents, because there must be damaging agents. Second thing, there must be any distortion inside the DNA, and what type of distortions are there, and what type of mechanisms are used to fix those type of distortions inside the cell. We'll see this all. Now the damaging agents could be X-rays or oxygen radicals or alkylating agents and all these things, or any kind of spontaneous reactions that are going on. Now as a result, what it can create the uracil abscisic site of 8-oxoguanine single strand breaks. So that means that it can create the single stranded breaks inside the DNA. So as a result of this X-ray radiations or oxygen radicals, which are very dangerous, you know, and alkylating agents, they all can create a single stranded breaks. That means we are having this DNA, and one of the strand is intact and another strand is have a nick in between the strand. It is called a single stranded break of a DNA. Now this kind of of break will be repaired by a system called base excision repair system. Now in this base excision repair system, what happens we will see in our future slides. Now in the second case, if we are having UV light radiation, now this UV radiation can cause to formation of the pyrimidine dimers, and most importantly they can lead to the formation of thymine dimers. Now as a result of the thymine dimer formation, there is a kink or huge bulky regions is formed in between the DNA strand, and as a result the DNA replication cannot pass through that region. DNA polymerase have to stop there. For this purpose it can create different troubles of DNA replication as well as transcription. So for this region we need to repair it, and we can repair it with a mechanism called nucleotide excision repair. Now both of them, the base excision repair as well as the nucleotide excision repair, are among the same family of repair systems. We'll be seeing why they are in the same family, but they are varying in the protein contents and different types of they are only varying the protein contents. The basic mean are the same, that means the chopping off of a nucleotide sequence and cleaving it away and incorporating new nucleotide sequence. That is the basic goal of all this repair system. Now in the third kind of damaging system we'll be looking at is via IR radiation or UV light or X-ray light, and whatever light would be there, and it can be anti-tumor agents. This can also attack the DNA strand, and it can create damages. Now the damage could be inter-strand cross-linked double-stranded breaks. So what it means, that there are double-stranded breaks. That means we are having something like that. This is the double-stranded break, or sometimes these are inter-strand cross-link. That means cross-linking of both two strands like that as a result of the recombination purpose. So we can have some structures like that. We can have this double-stranded breaks in between the DNA. So as a result of that, whole gene content will be lost, right? Because if we have the double-stranded break, the whole DNA will break apart and will be fall apart. So that's why we need to repair it as soon as possible, and the cell repairs it via the recombinational repair system. There are recombination repair systems, and actually this system requires the mechanism of recombination and some proteins of recombination to repair the DNA or double-stranded break of the DNA. I am not going to talk about this recombination repair system in this video, because I have already discussed this process in my previous video. You can go to my website, or you can go to my YouTube site and can find this the recombination repair. You can find a video onto it and go and watch it. Now and it could be any replication errors. Now during this kind of replication errors, what could be the errors? Like, as we know that A always try to pair with T and G always pairs with C. This is the actual rule of base pairing, but it could be transferred. For example, A can pair with G, T with C, and all this mismatch pairing is done. Or sometimes wrong incorporation of the base, or sometimes deletion of an important base can also possible during the replication procedures. Now for during this replication procedure, to block all this, we are having another mechanism which is called the mismatch repair system, or sometimes it is called the methyl mismatch repair system. It's also called the MMR system. Okay, now during this replication errors, I must tell you one thing. During the replication errors, fewer errors which are made during the replication are automatically fixed by the repair system utilizing the polymerase, which is the exonuclease activity of the polymerase. For example, polymerase start adding the nucleotide sequences onto the template strand to make the new strand. So it is adding all this nucleotide sequence. As soon as it incorporates the wrong nucleotide sequences, it shifts back its conformation in such a way that they can utilize its 3' to 5' exonuclease activity, and it will chew up few nucleotide and then again go on to synthesizing process. Okay, so this kind of system is called the proofreading activity of the DNA polymerase, and this feature is really important, because this feature actually give rise to the fidelity of the DNA replication process in much more detailing of the DNA replication process, and that ensures in the very first level that no mistakes are being made during this replication, or almost a very few mistakes are made during the replication. But still, right after that, if mistakes remains, in those cases we have to rely on the mismatch repair system or MMR systems. Now we'll be seeing all this later. Now another important thing I must tell you, utilizing this UV radiations, there are another mechanism. This is a very interesting mechanism utilizing the light. Now common light or sunlight have a higher energy component, as we know, because it's full of photons, right? So once there is a system inside the bacterial cell that they can utilize the energy from the photon of the light, and there is an enzyme called the photolyase which can utilizes this photon energy and can easily repair those thymine dimer formation in between the DNA sequences, and this process is called the photoreactivation repair system among the bacterial cells. We are also going to look at these mechanisms in detail. Okay, so these are the mechanisms we will be looking. Okay.
Although mistakes can happen during DNA replication, they are extraordinarily rare. A key reason for this is the proofreading function of DNA polymerase. Roughly 1 out of every 100,000 to 1 million bases attached to the 3' end of a growing DNA chain will be a mismatch, improperly base pairing with the complementary strand. When such a mismatch occurs, DNA chain elongation generally pauses. This pause allows spontaneous melting of the end of the DNA strand being synthesized, freeing the 3' end with the mismatched base to enter a 3' exonuclease site on the DNA polymerase. The exonuclease site catalyzes the removal of several nucleotides from the 3' end of the growing strand. Only the removal of the nucleotide carrying the mismatched base is shown here, though additional nucleotides would actually be removed. Following removal in the 3' to 5' direction, DNA polymerase can then resume its synthesis activity in the 5' to 3' direction. The proofreading function reduces the overall error rate to as low as about 1 in 100 million for DNA polymerase 3.
So what we can see that this DNA polymerase 3 are the exonuclease activities actually helping it to achieve this 1 in 100 million mistake in 1 in 100 million rate, right? So this rate of being rate of doing any mistake is getting lowered utilizing this 3' to 5' exonuclease activity by this DNA polymerase enzyme. That's really important. But right after all these necessary steps, if any mistakes remain, we need to depend on different types of mechanism systems. Okay. Now we'll be talking about this base excision repair. Now, as we know, the base excision repair systems are important because if there are damages by UV radiations, sorry, not UV, X-ray radiations and all this, it can finally leads to the lesions among bases, and this can be repaired by this base excision repair systems, and we'll be looking at in this animation.
Some damaged DNA bases are repaired by a system called base excision repair. Base excision repair works differently from nucleotide excision repair, which clips out a section of DNA containing a pyrimidine dimer or other defect to replace it with new DNA. Base excision repair acts on other kinds of defective DNA. For example, certain bases that are not supposed to be in DNA, such as uracil, are removed by base excision repair. If the base cytosine spontaneously deaminates, it forms uracil. Uracil can also be incorporated erroneously into a strand during DNA synthesis. If uracil forms in the place of cytosine, it would have a mutagenic effect. Whereas cytosine forms a base pair with guanine, uracil forms a base pair with adenine. During DNA synthesis, DNA polymerase would incorporate an incorrect nucleotide into the growing strand. A few other types of damage are also repaired by the base excision repair system. For example, when adenine becomes deaminated, it forms an unnatural base called hypoxanthine. A mutagen can also damage adenine by alkylating it, converting adenine to 3-methyladenine. Normally, adenine forms a base pair with thymine, but hypoxanthine in its place is mutagenic because it forms a base pair with cytosine. 3-methyladenine is even more damaging. When it is encountered in the replication fork, it stalls DNA synthesis. Consequently, repairing these mutations is vital for survival. Base excision repair employs a battery of glycosylase enzymes that can recognize and clip damaged bases from the phosphodiester backbone. Hypoxanthine DNA glycosylase, 3-methyladenine DNA glycosylase, and uracil DNA glycosylase recognize hypoxanthine and 3-methyladenine and uracil respectively when these bases are present in DNA. The glycosylases cleave the bond connecting the damaged base to deoxyribose in the phosphodiester backbone. The result is an intact phosphodiester backbone missing a base. The site is called an AP site. In this case, it is missing a purine and is therefore an apurinic site. If it were missing a pyrimidine, it would be an apyrimidinic site. The next step in base excision repair involves AP endonucleases that specifically cleave the phosphodiester backbone at AP sites. The 3' end produced by the AP endonuclease provides a starting point for DNA polymerase I to start adding nucleotides. At the same time, the polymerase uses its 5' to 3' exonuclease activity to degrade the cleaved strand downstream of the AP site. DNA ligase then seals the remaining nick and the repair process is complete.
Okay, now in this video I am very exciting about one particular point. Is about the activity of DNA polymerase, which is the 5' 3' activity. Now you know there are several activities like 3' to 5' exonuclease activity, 5' to 3' polymerization activity, and third one is the 5' to 3' exonuclease activity, because this activity can be seen in DNA polymerase I, as we all know this. Now that's very important, because we cannot see the activity of DNA polymerase I or the 5' 3' activity of DNA polymerase I in general process of DNA replication and all this. But in this case, in case of this base excision repair system, we have found the activity of this 5' 3' exonuclease activity in this case. Okay, so that's very important case. Anyways, now we have discussed about this base excision repair or BER. Now we'll be looking at the nucleotide excision repair systems. And as we know, UV radiations can cause damages like formation of pyrimidine dimers, and especially this kind of pyrimidine dimers are being get rid of via the nucleotide excision repair system. Let us see that. Okay.
Many microbes in the environment commonly encounter ultraviolet light, which can cause neighboring pyrimidine bases, thymine or cytosine, to form dimers. Thymine dimers are the most common. Pyrimidine dimers can be repaired by several mechanisms, including a method called nucleotide excision repair. In this system, a 3-subunit exonuclease, made up of the proteins UVRA, B, and C, excises a patch of 12 to 13 nucleotides spanning the dimer. ATP hydrolysis provides energy for many steps throughout the process. The repair mechanism begins with the association of UVRA and UVRB and the hydrolysis of ATP. The UVRAB complex binds to the DNA as UVRA makes the initial recognition of the DNA damage. UVRA bends the DNA near the damage. With more ATP hydrolysis, UVRA loads UVRB onto the damaged DNA, after which UVRA is ejected from the complex. Next, UVRB binds UVRC. UVRC nicks the damaged strand at 2 sites flanking the damaged bases. Another complex, called UVRD helicase, strips the resulting short, single-stranded damaged fragment from the DNA molecule. The single-stranded gap that remains has a 3' hydroxyl group that allows DNA polymerase to synthesize new DNA. After DNA polymerase is finished, DNA ligase seals the remaining nick in the DNA. Although this example showed the repair of a pyrimidine dimer, cells use nucleotide excision repair to fix a broad range of DNA lesions.
Okay. Now we have seen a very interesting process with the help of the UVR proteins. Now this kind of thymidine dimer can also be repaired with the help of photoreactivation mechanisms. We will be seeing the animation of photoreactivation mechanism in the next slides. Now let us move on to the next slide. Okay, now we have seen the nucleotide excision repair. Let me take a color first. Okay, fine. Okay, fine. Now we have seen the base excision repair, we have seen the nucleotide excision repair system, and in both of the systems what we can see, we see that all of the enzymes will come and act on the DNA strand, and it will cleave the DNA strand, some stretch of the DNA strand, and cleave it away, and finally which generates a gap in between the DNA strand, and this new gap is resealed by DNA polymerase. Okay, it is resealed by DNA polymerase. Okay, so this is the basic process. Now we'll be seeing a completely different type of repair mechanism, which is the mismatch repair system or methyl mismatch repair system, whatever you can say. Now this kind of repair system is needed due to make the repair between the incorporation of nucleotide like G in front of A or C in front of T. So we need to make sure that this kind of mismatches are cleaved out away, and the right nucleotide sequences are being added, and that can be ensured by this MMR procedure. Okay, let us see the methyl mismatch repair.
When DNA polymerase replicates DNA, it uses proofreading functions to prevent the misincorporation of bases. However, DNA polymerase does incorporate an incorrect base for approximately 1 in a million bases synthesized, resulting in a mismatch that distorts the structure of the double helix. Curiously, far fewer than 1 in 10 to the 6 mutations actually arise. The mutation rate in an E. coli cell is only 1 for every 10 to the 9 or 10 to the 10 bases replicated. The cell has a number of repair methods, including one called methyl mismatch repair to remove and replace a mismatched base. Methyl mismatch repair involves recognition of the methylation pattern in DNA bases. E. coli methylates its DNA at the palindromic sequence GATC using an enzyme called deoxyadenosine methylase or DAM methylase. DAM methylase adds a methyl group to the adenine in both strands of the sequence. Adenine and thymine bases pair normally. The DAM methylase acts soon, but not immediately, after replication of a DNA sequence. Where DNA has methylation sequences, the parental strand will be methylated, but the new strand will still be unmethylated. The difference in methylation allows the cell to distinguish the correct sequence, presumably on the parental strand, from the incorrect sequence on the new strand. A series of proteins called mut-S, mut-L, and mut-H act together to repair the mismatch. Mut-S first binds to the mismatch and recruits mut-L and mut-H. Mut-L recognizes the methylated strand and brings it in a loop. Mut-H then cleaves the unmethylated strand containing the mutation near the GATC. A DNA helicase called UVRD then unwinds the cleaved strand, exposing it to a variety of exonucleases. The result is a gap that is filled in by DNA polymerase I. The DNA polymerase continues through the region that originally contained the mismatch, inserting a correct nucleotide this time. After a while, the DNA polymerase falls off the DNA, and the gap that remains is sealed by DNA ligase. The methyl-directed mismatch repair proteins and genes are called mut because strains that are defective in one of these proteins have a high mutation rate. A bacterial strain with a high mutation rate is called a mutated